much. It is an incredible honor to be nominated for the Best Researcher Award by the International Research Awards on Network Science and Graph Analytics. Today, I am excited to share my research journey on the critical issue in sustainable agriculture. My name is Zeeshan Khan, and I'm a PhD scholar at the National University of Sciences and Technology at the Department at our Rahman School of Applied Biosciences. So here is our finding, and we published it in the esteemed journal, South African Journal of Botany, where we found that Mycorrhizopshere bacteria elevates the arsenic toxicity by regulating the organic acids, glycolase defense system, and the metal transporters in soybean plant. Along with my co-authors, I am really excited and really honored to be the part of such team, and we made it possible with the great efforts we made. To show you the whole summary of our experiments, when we made, when what we found in the control was when we gave the arsenic stress to the soybean plant, we found that the root biomass and the metabolic responses and the antioxidant defense system were really decreased, and the colonization in the roots were also decreased the as arsenic was the toxic element for the microorganisms to get into their symbiotic relationship with the soybean, and through that the plant experienced the diverse stress, and the whole of the activity of the plant was decreased. Whereas when synthetic community and herbuscular mycorrhizal fungus was inoculated in the combined effect, which we found the best result of our study, and we found that all the metabolic processes and the antioxidant defense system and all the root system activity were uh, proceeded to the normal, and ultimately we also found that the root biomass was increased. So we found that the synthetic immunity along with the beneficial fungus can be a sustainable approach in for the agriculture practices which can be made, and to cope up with the other needs we need in the present time, like we need to also lower down the effects of the chemical pesticides, or we gave the spray to the agriculture sector, agricultural lands. They are also causing damage to the human health and ultimately the plants too. So we need a sustainable approach, and what we think and what we came across from this experiment, that this synthetic community and AMF is a sustainable approach to not only cope up with the heavy metal stresses, but it will also increases the health of the soil and ultimately to the health of the plants. So moving on to our first finding, as I told you that there were we gave we were having two controls in our study in which haven't placed any, sorry, three controls, haven't placed any uh, stress or any inoculation. Then we gave the combined inoculation to check what are their effects if no stress is placed in the soil, and how the plant responses to it, and then the arsenic stress, and then we get synthetic immunity to check their individual responses and to the heavy metal stress, and then the combined response to the heavy metal stress. What we found that these mycorrhizas bacteria. To move on, first of all, I guess I should let you know that synthetic communities are the one with which we made a cocktail of different bacterial species, which are having the synergistic effects to each other, and they are beneficial for the plants and even to the soil. And the purpose of using the MF is that they got the immense potential to recruit the major microbial organism from the environment to the environment of the plants. And then, okay, so move on to this section. we found that mycorrhizophere bacteria, they enhance the soil growth, plant growth via different mechanisms, which will be explained in the later slides. And all the photosynthetic activities and the biomass were increased in this majorly in the combined uh, inoculation of synthetic community and MF. Then we found that root colonization was increased when combined um, mycorrhizophere bacteria were placed, inoculated, sorry. So what we found that the colonization was increased, and when the colonization is increased, so the colonization will increase the uptake of the nutrients to the plant, and ultimately the plant health will be uh, increased. So it is it is also the hypothesis till now that they are that the bacteria or the fungus, they um, use their reside on the heavy metals, and then they use them as a substrate for their energy. Might be it is possible they are interacting on it and placing some biofilms on it, and then the lower effect is placed. Moving on to the third section, mycorrhizal bacteria, it alleviates the oxidative stresses as when arsenic stress was there. So the oxidative stress was increased within the soybean plants, and we can see in the section in which arsenic increased the hydrogen peroxide, LOX enzymes, MDN, and methyl glycolide Mg, and which is ultimately the biomarkers that oxidative stress was increased when the arsenic stress was placed within the soil. So when they are present in the simple or in a normal quantity that as a biomolecules, but when synthetic communities and MF was placed within the arsenic stress, so they normalized these um, oxidative stress markers, which when oxidative stress markers are present in the high quantity, they actually disrupt the cellular machinery of the plant. So the synthetic community and the arbuscular mycorrhizal fungus, when inoculated combinedly, they lower down the effect of this uh, oxidative stress markers within the plant, the arsenic stress, and then when we see the antioxidant systems, yeah, they were elevated when arsenic source stress was there, but when we placed a cocktail of our inoculation, so they actually increased the antioxidant defense system, because as you know, as we have seen that in the they have increased the colonization within the roots, so might be they increase the uptake of the nutrients from the soil, and ultimately they releases some type of metabolites which increase the antioxidant defense system of the plants, and they also when we see that organic acid production was also increased within the plants which were placed in the arsenic stress condition. So actually, organic acids are the one which are being released from the roots to cope up with the stress condition, and ultimately we found that, or in our study, that synthetic community and vascular mycorrhizal fungus, they increased the organic acid production within the plant. So this shows the healthy nature of the plant. And our last finding was that they also increased the expressions of the metal transporter genes, as I already told that the uptake of the nutrient from the root was increased when we placed our cocktail. Then we found that the different regulations of the heavy metal transporter genes were also increased within our um, inoculation. And to conclude all of our study, we found that the synergistic action of the synthetic chemical and MF, it significantly mitigates adverse effect of arsenic, and it enhances the plant physiology, and ultimately it is the best uh, source that we count on for the sustainable optical strategy. Thank you so much.